Thank you again for inviting me. I did not intend to give a talk, but I was very happy to get the invitation. So I'm happy to be here. But yeah, so not only am I the paleontology program manager, but I'm also the museum curator at the park. And so that means my responsibilities are the fossils, the rocks, and everything that's in the museum collection. And our museum collection includes art, archaeology, the fossils, a wide variety of other things. So there's a lot that we manage in our program. And I've been at John Day Fossil Beds for about 5.5 years. I actually was hired by the National Park Service before I graduated. Not by very much time, but it was hard finishing my degree and then having to come back to my university and stay in a hotel and defend my dissertation, but I did it. Okay. So first thing I'll mention too is that, so I work for John Day Fossil Beds National Monument, which is a unit of the United States National Park Service. And the National Park Service is part of the US Department of the Interior. And the Department of the Interior is underneath the President of the United States under that branch of the government. Yes. And so I wanted to do a little bit of explanation as to what exactly it is and where we work. So I am where the red star is here in the National Park Service, which is under this assistant secretary, which is under the departments, or the deputy secretary, which is under the secretary. And what's cool is, for me, our secretary is Native American, and she is the first Native American Secretary of the Interior in the entire history of the United States, which is important because you'll notice right here is the Bureau of Indian Affairs, which is our branch to interface with, which is interesting that it's in the Department of the Interior. We have the division to work with the Native peoples. And some of our partners that we work with, who are also federal agencies, include the U.S. Bureau of Land Management, the U.S. Geological Survey, U.S. Bureau of Reclamation. So there's a lot of different offices that we work with. And so this is just a way to show that if you work with any of us, it's rarely ever going to be the same experience. It's very different depending on which agency, which secretarial office. And then one that's not on here is the United States Forest Service, which is another big one for paleontology in the United States. So again, these are some of those agencies a little bit more specifically. The ones with the yellow stars are agencies that manage land. Well, I should say the bold ones are ones that manage land. The ones with stars are the ones who have paleontology resources that they work with more frequently. And Bureau of Indian Affairs does have fossil resources in it, but we don't work with them very often. We usually work directly with the tribal governments when we have to work with them. Now, the National Park Service itself, it's broken up into many different regions. So there's many different subdivisions. So we have a branch called the Washington Area Service Office, which is the national office. So that helps us all across the country. And then each is broken down, each part of the country is broken down into regions. So we have the one that I'm in. So I'm up here in this state is Oregon. Oops, sorry. I touched it. Oh no. So I'm in the Pacific West region in the state of Oregon. Now, Pacific West region also includes the Hawaiian Islands, American Samoa, everything in the ocean. So we have everything in the Pacific, Oregon, Washington, Idaho, Nevada, all managed as one region. Whereas everything in the Washington, D.C. area is managed as one region. So then there's a lot of different things about this. And we recently changed these regions to this new system that has all of these numbers, and it's supposed to be based on the way that the rivers break down, and it's very confusing. And made life a little hard. One thing I find funny is there's this big chunk out of Oregon right here, and that's where Crater Lake National Park is, the only national park, full name, in the state of Oregon, and it's in a different region from all the other states, everything else in the state. It's very weird. And that's actually another good thing to talk about. So then we get down to parks themselves. So parks is a term that we use to describe each of the units of the national park system. So I work, even though it's national monument, I still call it a park. So that might be confusing because we are all the national parks, but it includes things like seashores, parks, monuments, variety of different things in there. So that's kind of what that means. And then there's another separate thing called this INM stands for inventory and monitoring. And that's usually what our ecologists work on. So they go in and they are working on the plants and the animals and whatnot. They're doing a lot more of the science because the National Park Service is not an agency that typically works with science. Our job, the mission of the National Park Service is to preserve unimpaired the natural and cultural resources of the national park system for the enjoyment of future generations. That's what we're there for. So primarily we're conservationists is what we do, but we also allow people to come and play, which can make things hard in a lot of different ways. And then there's also specific programs that are kind of strewn throughout this. And so if you worked with the National Park Service, you could potentially interface with any one of these structures in any different part of the country, and every interaction might be different depending on who you're working with. In the national office, I do work with some people called in the Geologic Resources Division. We also have a Biological Resources Division. We have an air quality program too. There's a lot of different programs that we work with in the national office. Another funny thing, Alaska on its own is its own entire region, the only state that's its own region, which makes sense. It's the biggest state in the country. Okay, so this is a fun map. This one shows every yellow dot is a site where there are fossils, a National Park Service site with fossils in it. Every green spot is just a National Park Service site. So as you can see, it's a vast majority of the sites have them. So there's 276 units with fossils, and there's about 423 sites total throughout the units throughout the entire Park Service. I'm up here in this part of the country, and I've worked over here at Badlands National Park, and I've helped with resources in what's that one called again? Channel Islands in Los Angeles, as well as down in San Diego, down here. So, but all of these places potentially have them, ranging from very old, from 500 million years ago, all the way up through 10,000 years ago. We have all of those represented. There's even in Texas, there's a very cool fossil site which has a herd of mammoths, females with calves. It's the only one known. It's a very cool site. Waco Mammoth Site. Let's do this. Maybe. No, okay, didn't like that. Now it doesn't like it. Fine. So then this, what the red stars here are representing are where there are paleontologists whose job it is to do paleontology all the time in the Park Service. So of all of those sites, these are the only spots that actually have people like me. Now, there are other paleontologists that work at various sites throughout the country, but their primary job is not just paleontology. They will work on many, many other things. Like there's some people in the Las Vegas area over here that they have paleontology training, but they do archaeology and all these other things too, because they have to. Or some sites don't have anybody at all. And so there's a lot of sharing that goes on. Like over here, Badlands is a shared position with some of the parks here in this part of Nebraska. So we get pulled in many, many different directions in that case. And we do have someone in the national office that tries to help a lot of these other sites throughout the country and coordinate as best as he can. And we also have partnerships with the Society of Vertebrate Paleontology and the Paleontological Society. Okay, now I'll actually start getting into why I'm here to talk about my science. So in the shadow of volcanoes, we know that volcanoes have a lot of direct impacts on life. Ash is distributed out, which is a barrier for life to get to, for plants to get light and for animals to get clean food into their systems and clean water. And sometimes you get, like in this case in Mount St. Helens, just knocking down and killing everything right in its path. So there's a lot of these direct impacts that we find from volcanoes. We know this. This is very well documented all over the world. And we see a lot of variety of devastation that happens. We see removal of trees, burning of the land, pyroclastic flows, which I like to refer to as fiery clouds of death, right? They come across the landscape and just kill everything. Fluvial deposits like mud flows, and of course lava flows. All of these impacts change the ecosystem one way or the other. Now, what do we know? There's a long history. Volcanoes have been on this planet longer than life has been on this planet. We have been evolving with it forever. Generally, everything that's directly in the area we know dies. Many things come back, and geography matters. Where you are in the world matters. A volcanic eruption closer to the equator will have a different impact than, say, one up in Alaska. And we know a lot about fish and birds and plants, but we don't know a whole lot about mammals. And mostly what we know about fish and birds and plants is thanks to Mount St. Helens in the state of Washington. Once that eruption happened, everybody got excited about volcano ecology and wanted to focus on it. And there have been some other places, some other volcanoes where we've worked on, but they're usually islands. So that's another place we don't know a lot about, is continental eruptions. As far as ecology is concerned. And mammals are hard to do because they don't like to be caught. They're a lot harder to catch too. And so what I've been looking at is specifically ecology. So this is an example of what's called succession, right? And so you have an eruption, and then there's a certain amount of time that it takes to add new species to the area until you get a viable population again, a stable ecosystem. Another area that I looked at for this study was morphology. So you have these are fossil horse skulls from Oregon, and the question is, what happens when you have a volcanic eruption? Do the animals stay the same? Do they get bigger? Do they get smaller? Is there any change in their shape? That was one of the questions I aimed to look at. And one other area I'm going to briefly touch on is reproduction, the reproductive strategies, things like rodents, which produce a lot of offspring without a lot of parental care, versus animals like elephants and humans that invest a lot of time in their children over a long period of time. So what is volcano ecology? Volcano ecology is the study of the interactions of volcanoes in the ecosystem. This is an example, a cartoon example from Mount St. Helens with an adorable gopher popping up after the eruption and saying, "What happened to all of my food?" Right? And then this is another artist's interpretation of one of my favorite sites in the central part of the United States called Ashfall Fossil Beds. This is a site that has 300 complete skeletons of rhinos and horses and various other animals, all preserved three-dimensionally in the ground. It's been referred to as the Pompeii of the United States. It is an absolutely gorgeous site, and if you're ever in the central part of the United States, I would highly recommend going. It is absolutely marvelous, this site, and the preservation that you get there. We can learn so much about what happens to these animals in these situations too. I don't talk about it very much. I'll just briefly talk about it here though. We know from this eruptive event that the animals were breathing in the ash and dust, and that it caused damage to their system where they developed a bone growth disease. And we can tell that the animals that lived longer had it worse than the ones who did it. So the smaller animals, the animals about this big, had no bone growth, and the rhinos had a lot of it. So it was very obvious that the longer the animal was alive, the more they were suffering from it. Also interesting, the baby rhinos did not have the disease, but the adults did, which suggests that because the babies were still feeding from their mothers, they weren't digging through the ash to get to the grass. It's a really cool site. You can learn so much about all life from that ecosystem. But at any rate, so when we have an eruption, we have a primary disturbance that changes your pre-eruption ecosystem. And it changes a lot of different things. You change the types of animals, the soil, the organic matter, and then you get new deposits sitting on top. And this is the first step. And if you don't have another eruption, you'll have a dispersal of other organisms from other places that come in to kind of repopulate the area. And then if there's a second disturbance, you kind of start it all over again. But if there isn't one, then you just keep accumulating more species through time. And this is the general system of how volcano recovery works. This is an example of succession. So these are two examples of birds. So the black line is Mount St. Helens. The gray line is Surtsey, which is an island off of Iceland. And what is interesting is this is from the volcanic event on the left, and then it's years on the x-axis, and y is the number of species. And generally speaking, it looks like the birds, there's a couple of fluctuations, but generally about 10 to 15 years later, things are starting to look kind of good, but it does take a long time. It took until 2010, and the original event was in 1980. Whereas with Surtsey, it was just a constant growth as the birds found it, though it took them about 5 or 6 years to finally find the island in the first place. But eventually they all got the memo and they started hanging out there. So just some examples of what that accumulation looks like. So modern volcanoes, if we want to understand volcano ecology, we need to look at modern volcanoes. And there's a lot of different ones that we could use that have erupted fairly recently since 1980. This is a short list here of the different ones, some of those examples. But as you can see, many of these, well, not all of these have erupted since then, but these are all examples that we could use. Many of these are islands. Islands are useful, but they're self-contained in a way that continents obviously aren't. And so if we really want to understand continental, we need to focus on continental. So I specifically focused on Mount St. Helens and also Mount Lassen. Mount St. Helens is in Washington State, and Mount Lassen is in Northern California. And so why did I pick these two? They're both continental, so they're comparable to one another. Mount St. Helens is a very well-studied volcano. We care a lot about it since that original eruption. There's a lot of data that's available for it. It has a lot of volcanic data as well as biological, so we know pretty much every in and out of that particular eruption. We have a really good pre-eruptive data set. We know fairly well what was living there beforehand. We also know a lot about what was living there right after it, approximately 5 or 6 years after the eruptive event. Mount Lassen is a very different eruption. It's a much smaller scale eruption. It happened in between 1914 and 1917. And so we have approximately a century level scale that we can look at to look at those relationships. Obviously, it's much smaller eruption. It was basically just mud flows, whereas Mount St. Helens was a very big, massive event, comparatively so. Obviously, there have been bigger ones in history, but it was a fairly large one by recent standards. So how do we know when we look at that ecology that it's really the volcano and not the environment, the normal environment, the changing climate? Well, we can actually use a control system, which for a paleontologist is very rare and very exciting. So I used Mount Rainier and Mount Shasta as my two comparatives. So Mount Rainier is in Washington. It's within about 30 kilometers of Mount St. Helens. And I also used Mount Shasta for the same reason. It's about the same distance away from Mount Lassen. And so they should be, and I chose 30 kilometers because that would have been outside of the blast radius of the volcanic eruption. So there should be no impacts on that volcano. So we'll look a little bit at some of the data here. So Mount St. Helens, this graph here shows time on the x-axis and richness, so species richness on the y. This is a methodology called chow richness, which calculates an estimation of, calculates an error, as well as estimating the number of species based on the number of specimens that you found. So I see this as a way to sort of account for any missing specimens, any missing animals, because you can't catch every animal. So this is just an example of trying to get at that. And so what we notice is these are our pre-eruptives. The red line is Mount St. Helens. The blue line is Mount Rainier. So blue is our control. And what we notice is that we do, in fact, have a much higher richness before the event. Our little cartoon there is marking the event. And immediately after the eruption, things are much lower. And then it does continue to drop, but we do see what looks like succession going on with Mount St. Helens, where there is this increase through time, generally speaking, especially given the error. Whereas our blue line, our control, is not really doing the same thing. So if it was a matter of climate, I would expect the blue line to do the same thing as the red line, but they're not really doing the same thing, which suggests that this may be something going on with the local ecology and dispersion into the area. So what we can tell from Mount St. Helens, it looks like there's short-term recovery, and it does look like there is some succession. We're looking at maybe about 5 years before we're pretty close to what we had before. Now, if we go to Mount Lassen, we notice a very different pattern here, where and I should mention here that and this is the same sort of graph with time on the x and species richness on the y. These points on the end are bounded by massive field collections that were done by the University of California. There's 3,000 specimens, I believe, in each of those bins, 3,000 here and 3,000 here. So we know that that's what's in those areas. There's no way, like, based on any other collections, and that's why these error bars are so small. But the blue line is doing some weird stuff also. So what this suggests is that species richness is relatively even over time through this particular event. And with both, we're generally seeing a decrease in species through time. And yes, there is this weird bubble up there, but most likely the reason why that happened was because of targeted field collections. So it wasn't, let's collect everything. It was, I want exactly this. And so when you go out to target specific species at different times of year, you can artificially increase the number of species that are present at any given time. And so what we see is a fauna that appears to be relatively stable over 100 years. Climate is playing more of a big role in this particular case. And maybe this eruption was just simply too small to matter to the overall environment. Now, this is an interesting plot. This is an NMDS, which is a non-multidimensional scaling. And so basically what this does is it takes similarity or distance really difference, and it plots it in a way that kind of visually makes some sense. And so what we see is, so the orange is Mount Rainier, the red is Mount St. Helens. So you've got these hot colors for Washington. I should have flipped them around. I should have had the hot colors be California, where it's hotter. But the blue colors here represent California, with these lightest blue being Mount Shasta and the darkest blue being Mount Lassen. So this is Mount Lassen is our volcano of interest here, and Mount Rainier is our volcano of interest. Or sorry, Mount St. Helens is our volcano of interest over here. The first thing that we see that is very nice is that California and Oregon or California and Washington are very distant, which means it's working. They should be. There's a whole state in between, so that's pretty good. But what is also interesting is that pretty much the cluster over here, the California ones, don't really change all that much. That 130, that middle point that was off differently, that's the one that's different. And as I mentioned before, that turns out to be an artifact of how they were collecting during that time. Whereas all of the other sites, and yes, the Lassen and the Shastas are close together, but they're closer to one another than anything else. So not really seeing a whole lot going on over there. This particular one, the Washington one, is where things get very interesting. Your pre-eruptive event at Mount St. Helens is very different from everything else. And then you have this volcanic event, and what happens? Well, it looks like that the post fauna of Mount St. Helens is more similar to what we see at Mount Rainier, both pre and post, which is suggesting that you may be getting these source populations from places like Mount Rainier coming in and repopulating the area. So this is interesting. So what did we learn? Scale certainly does seem to matter. Bigger eruption like Mount St. Helens does seem to have had a bigger impact than the smaller Mount Lassen eruption. There's some evidence of suggestion, or of succession, if I can enunciate. There does seem to be changes in the players, migration of neighboring populations, those source populations into the area. Climate still seems to be playing a role in certain areas to some degree. And there generally does seem to be a quick turnaround with about 5 to 10 years, at least with these size of eruptions. But that's only 2 examples. How can you say anything about 2 data points? That makes a line, right? That's not useful. Where else can we look? Well, can we cause eruptions to happen? Well, they certainly tried during the Second World War. This is they tried to blow up some volcanoes in Japan. Did not go very well, doesn't work. And as a National Park Service employee, I can't advocate for trying to set off a volcano. That's a terrible idea. So we could wait for something to erupt. And we do have fairly good ways of predicting when volcanoes will erupt, but we don't know that far in advance when an eruption is going to happen to collect the data that you need to answer these questions, because you need a really good pre-eruptive fauna as well as a post-eruptive fauna. And like I said, we can examine the volcanoes, volcanically active areas, but we just don't know, right? We could go and sit on a volcano here, right, in Europe, in Italy. We could sit there for a long time before we can expect any of those to go up. Right, yes, we don't want those to erupt, right? Especially in some of those places where they are, that'd be very bad for a lot of people. But what else can we do? Well, we can look at the fossil record, right? Things that happened millions of years ago, which is wonderful that we could do. And here are some examples. So ashfall pops up again. So ashfall fossil bed, this is an example here. Gorgeous, 3-dimensionally laid out horses. Absolutely beautiful. I love this place. This is so cool. And this is about 3 meters worth of ash. Just solid right there. So sparkly and gray. It's gorgeous. Absolutely. I could go on about that forever. This is another cool site. This is called the Blue Lake Rhino. This is a rhino from, and I should also say this site is about 12 million years old. This site, the Blue Lake Rhino, is 16 million years old. And this is a rhino that was captured in a lava flow. Most likely what happened is this animal died and was in a lake, and then the basalt flowed into the lake and covered it over. Reason why we estimate that is that the rock around it is what's called a pillow basalt, and pillow basalts only form in the presence of water. So that's why it's a very fun rhino-shaped hole. And we know, in fact, it was a rhino because there were rhino bones found inside of it in the 1950s. And this is up in the state of Washington, and those fossils are actually down at the University of California. But so these are some examples. There's about 1% of all fossils are found in volcanic rock. So and I say like, well, there's not a lot, but still 1% of all fossils is a lot of fossils. And some other places we can look at includes, there's this really cool rhino that was found in Turkey. This is in what's called an ignimbrite, which is one of those pyroclastic flows. So one of the fiery clouds of death I had mentioned before. And then we also have where I work, which is the John Day Formation. And granted, this is not what I have here. This jawbone of a mouse deer is not in a volcanic layer, but this rock has a lot of volcanic material mixed in with it. So it's what's called volcaniclastic. And there's so many, and I will explain how many there are here in a second. So if you're not familiar again with where we're talking about, Oregon is this chunk of the United States on the West Coast. And specifically within Oregon, this is the area that we work in. The purple dots, which are a little hard to see, those are the units of the National Monument that I work for. So we have those three chunks, and that's all we really cover. And the green square represents all of the area that we have collected fossils from as John Day Fossil Beds National Monument. And this is not even all of the areas where you can find fossils in this part of Oregon. It includes pretty much everything out here of the kind of fossil that I care about. There's also marine fossils and all these other things all over the state. It's a very diverse state. But for my purposes, we focus on these areas. And specifically for my study, I was in where the red square is, in the sheeprock unit of the monument and a little bit outside of it on some partner land. So here is the stratigraphy of where I'm working. So it's about 400 meters worth of rock. And within that, all of these green layers are the volcaniclastic sediments I talked about, and each of the gray layers are actual volcanic layers that we can get an age on. There's approximately 14, I believe, total, and this selection of dates is not the complete list. We've developed even more working with our partners at Boise State University. And we probably have about 30 to 40 total throughout the entire monument that we're getting new dates on, which have about 10,000 year errors, which is outstanding. I know that may not seem like a lot to people who work in the modern system, but 10,000 years is a lot. It's amazing is what I mean. It's a very short amount of time. So we can get very precise dates with all of this. And this is an example of what that looks like. This is a sheeprock. It's a big mountain that I work in a lot. And what I also find very interesting about this site is specifically this very thick gray layer. It's called the Picture Gorge ignimbrite from about 29 million years ago. It's this big brown strip right through the middle of the face there. And I think we're looking at maybe 50 meters or something like that worth of material. It's a lot. And the estimation is that volcanic layer, before I go on to that, the volcanic layer is a very big eruption, and I'll explain a little bit about it in a second. But the other thing to keep in mind is that the way that the Turtle Cove member of the John Day Formation is broken up is by these letters. So we have A, B, C, D, E1, E2, E3, all the way up through a subdivision, 2 subdivisions of K. And that's how we break it up. I jokingly call it an alphabet soup. But so that big brown layer I talked about is from this big red spot in the center of Oregon called the Crooked River Caldera. And this caldera is something on the order of maybe 45 kilometers wide. Right? It's huge. It is a big hole, and it is a caldera forming eruption. So when this thing went off, it would be like if Yellowstone were to erupt today, right? Big area with just stuff coming up everywhere. Absolutely horrible. And that's what we suspect that volcanic layer came from. So we do have evidence of a supervolcanic eruption in the monument, which is wonderful that we can look at that. Not so good for the animals 29 million years ago, but this is an important tool that we're actually able to use. And these gray layers represent the areas where the John Day Formation is exposed. So there's obviously a big section here that's subdivided by that eruption, that eruptive event itself. And again, we're focusing over here on the sheetrock unit, and granted that that's about 100, 150 kilometers away, it's still very thick. So it was a very scary event. So then we just jump right into the richness again. Each one of these represents that alphabet soup of layers. So you're going into the future here. So we're over here in the years that way. But this would be A, B, C, D, E1, E2, E3. And then each one of these cartoons is representing some of the eruptive events. This big eruptive event is representing that big ignimbrite. And what we can see is that roughly it stays pretty constant through time, except for right here, it starts to increase a little bit, but there's a big error on it. And then it drops and then starts doing the same sort of increase that we see like we saw with succession. Oh, that's interesting. And it could be a few things, but also keep in mind that this dot here is very narrow on the error, which means there's a lot of specimens to back up that spot where it is. So we definitely do see a drop. And then it does appear that even if you look at, even if you squint a little bit with the error bars, there is an increase through time. And then this is looking at that non-dimensional scaling again. And I left all the species on there in this particular plot, so it's a little bit messier than the ones I showed you before. But you can still kind of see that there's three major clusters going on there. Everything here is before that ignimbrite. This one is immediately after, and this one is later in time by about 2 million years. Right? And that was actually something else I didn't mention before, is that the base of the Turtle Cove member is 31 million years old, and the top is about 28. So we're not talking about a huge amount of time, geologically speaking. This is relatively instant for me. So then again, what this is showing is that there are specific species that kind of push these different layers in these different directions. And if you could read these a little bit better, what it would show is that most of the animals that are pushing in these directions are rodents and rabbits, things that are open habitat animals, whereas everything that's over here are closed habitat animals. You see a lot of animals that show adaptations for living in and among trees. The horses are very different, but I'll get into some of those numbers a little bit different, display it a little bit differently here shortly. So who are the players? Who exactly are the ones that are changing? So this graph shows the different families that we have. And this is a relative percentage of that particular layer total for each family, with the stratigraphy here, starting with the oldest on the bottom and the youngest on the top. And so some of the key ones to pull out here, the red bar represents that ignimbrite, that big eruption. This one, these are mouse deer. These are hypertragulids. These are very adorable, about the size of a loaf of bread sort of animals. And they were very common up until about that event. Where do the animals like that today live? They live in Southeast Asia. They really like forest and jungle, which I tried to combine into one word. That was weird. So that's a forest or a closed habitat group. Mericoidodontidae, these are oreodonts. This is an extinct group. I like to say that they're sheep, camel, pig-like animals. They're more closely related to camels than anything else, but they don't have any modern relatives. They show adaptations for living in closed habitats more so, at least the ones where I am. Dogs, the canids, they're doing really great. And then, I mean, there's upwards of 12 co-occurring species. which is unheard of now in one spot, right? They did not do very well after that eruption. They were not. Rhinos, same thing, not very common afterwards. All these animals really liked closed habitats. These particular ones did. But what's common afterwards? What are really taking off? The Apodontidae, which Apodontids are mountain beavers. So they're a rodent. Leporids, the rabbits, Chryseidids, Geomids, all different groups of rodents. They're doing very well. So what I think is going on is that we see the eruption go off, knock down all the trees, open up the habitat, and then these other organisms are starting to come in to fill in those gaps. Right, so kind of to visualize what this looks like, this is our pre-eruptive environment that we see with rhinos over here, horses. These are our three-toed horses, the oreodonts, lots of dogs, false saber-toothed cats, a few rodents, our cute mouse deer, and this thing called an entelodont, which is a hell pig, Terminator pig, very big animal, big omnivore, very interesting. And then you have the ignimbrite and snap. More than half the population is gone. So things become less common. You lose a lot of things, but then shortly you start getting rabbits back and the horses start to come back. So we're seeing some succession shortly after. So what did we learn from the John Day Formation? Size really does matter in this particular case. Massive eruption causes massive changes. Climate is still important, obviously, though, and I didn't mention this before, but climate is pretty stable through time during this period of time globally. But there seems to be an impact from that eruption. Now, I'm only going to briefly talk about these other bits here because I don't have a lot of data to show, but shape and size, what happens there? So that's wrong. No, that was the right one. My bad. So in South America, one of the cool things that we see there is this adaptation towards these more trunk-like structures to form on some of the animals. This is a horse with retracted nasals. You'll not see a horse like that in Europe or North America. And what this seems to be an adaptation for is a very fleshy lip, sort of more like what some browsing animals have. I don't remember which rhino has that. I keep getting it. The white rhino has a very muscular lip. And what's interesting is that animals that tend to live in very volcanically active areas do tend to have these muscular lips for grabbing and stripping off leaves, it appears. And also there is a group of primates that does appear to do this in Africa, where they have actually very grazing adapted teeth, but they pick the leaves and strip the dust off and then eat them. So there does appear to be a similar structure like that. And the other thing that we'll notice is the height of the teeth. So animals that live in abrasive environments have much higher teeth, taller teeth to deal with all of that extra stuff that they're working with to break down their teeth. And what did we see in the John Day? I looked at to see if there's any size difference that occurred. And there was no size difference there. So what's probably happening is that when the population of horses is killed off, a new population is immigrating into the area and there's enough resources there that they don't need to change their morphology. So, yeah. And then very briefly, I just wanted to mention reproductive strategies. So, versus what has been termed the R selection versus K selection. R selection being like rodents producing lots of offspring, and K selection producing very few but with a lot of parental investment. I think I have a slide here. Yes. So you see an increase. So what the estimate is that we see an increase in R selection versus K selection immediately after the event, the volcanic event, and then as time goes on, you get to see that. Now, I haven't actually had an opportunity to look at this with modern systems yet because I didn't have an easy way to quantify that to actually test it. But Edward and I and Sam Hopkins, who was here earlier, we worked together and we actually developed a methodology for quantifying reproductive strategies in organisms, in mammals specifically. So we have this wonderful system using the reproductive strategy index that we developed. And we did some work testing that, to see if it actually made sense. So we did it with body mass and diet to see if there was any sort of relationships. And generally speaking, it did work. There's a lot of interesting things we were able to pull out of that. It is controlled by phylogenetics, so who you're related to does really matter in this particular case. But the other thing that also seems to matter is your locomotion strategy. So if you are flying like bats are, you do weird things compared to other animals of your same size. And whales, which are technically carnivores, act more like herbivores in this system because they're bulk feeding just like a cow would, right? Just a very, very big cow. So we can learn a lot about, so we've learned, I should say, a lot about reproductive strategies doing this, with the future application of this system being to look at the volcanic systems and see, do we actually see that same relationship of things that are more R-selected showing up immediately after the event and then transitioning to a more, a wider distribution later. How long does it take for those case-selected taxa to show up? These are the questions that we want to ask, and now we have a tool to answer them if we could ever find the time to answer the question. There are so many questions to ask. General conclusions. Volcanoes produce a lot of amazing fossils. Scale of eruption is very important for ecology. Bigger eruptions it means that it's longer until stabilization. We see evidence of succession through these events like we do with many other sorts of events, ecological devastations. You see these migrant or immigrant populations coming in from a source population, obviously playing an important role. The morphology bit is complex and the reproductive strategy is interesting, and we definitely have room to learn a lot more about what's going on in that part of it also. These are all the people I would like to acknowledge, and of course I forgot to acknowledge you all, but thank you. And that's all I have. Thank you. Oh, the black rhino. Okay, thank you. You made the same mistake I did. I can't remember. It's because I'm North American. I'm not used to the rhinos. We barely even have them in our zoos. So
I have a question about
yeah.
So I saw all the volcanic events, the John Day and the big one, the event. Does the others, could you measure if the others had an impact?
Yeah, so let me go back to it if I can.
Quantify the difference between the big one and the smaller one? That one.
Yeah, I probably could. I haven't done a lot of work to do that yet, but that is something that I want to be able to ask, especially now that we have more volcanic events that we have good dates for. But it does get a little difficult just because of a lot of the confounding factors that are involved with the smaller ones. So I'd be a little bit hesitant on the smaller ones. But I definitely could do that. And that's another area, especially if I had a student who was working with me on this, I think it would be really interesting to work on a method for trying to quantify that, or finding a way to quantify those exact differences between the sites and how exactly they change.
Do you know the size of the smaller eruptions? Are they comparable to the ones lesser?
They're all bigger than Mount St. Helens.
Okay.
So a lot of these other ones are volcanic layers that are about this thick, whereas Mount St. Helens, for example, is about that much.
Okay.
So I think, if I remember correctly, what it was like 2 or 3 centimeters, or not even that many. It was like maybe 1 to 2 centimeters, I think, for. Right, yeah, there's no compression for Mount St. Helens, but then these events have all been compressed and you're looking at 1 meter in some cases. So these are much larger events, and those small ones would be almost impossible to identify once they've been compressed. I don't know what the rate of compression is, but so at any rate, to answer your question, they are very big eruptions. I don't know the exact size. In fact, most of these eruptions, we don't even know where they came from. We know where this one came from, but a lot of the other ones, it's like somewhere in the Cascades, somewhere in the mountain range at the subduction zone, maybe. Or it could be this other hotspot location in other places, but so we don't know.
Are you trying to figure out where they come from, or?
There are some geologists who are trying to figure that out. It's not as important for my questions to know exactly where they come from, so it's not been a high priority. But it would be nice to know where they're coming from, especially in trying to understand how much of an impact they had, because it may be that these are so thick because the volcanoes were very close, right? And so knowing that proximity would be really important for my study, but
yeah, you can not put your time.
Yeah, exactly.
And is this based on species or in general?
Yes, this is, I believe I based this on species.
Okay.
And also keeping in mind, which I know many of you probably are aware of this, but fossil species are not the same thing as modern species. Yeah, so yes, but they are based on species. And so that has been one of the things that I've been working a lot on. Why I haven't published a lot of this yet is trying to make sure that we have as close to scientifically defensible species as possible. Because I mean, even just working on those mouse deer, when I started this project almost 10 years ago, we suspected that there were 3 species of mouse deer, and doing subsequent work has shown that there's probably only data to support 1 species, at least from the bones. It was supposed to be 3 species, 2 of which were in 1 genus and 1 was in a second. And when you look at the variation of that same population versus a modern distribution with the same sort of breakdown or taxonomically, 1 single species of modern tragulid has more variation in it than all of the hypertragulids, the fossil ones combined from the 3 different species and 2 genera. So who knows what we're really looking at there. But yeah.
And how many species are in average each?
There's about,
just to have an idea of the richness.
Yeah, so the estimated values look like they range from about 30 or so to the one at the end there has almost 110 species, but most of these are about between 50 and 70, so probably an average of about 60 species of only mammals. Yeah, and I didn't mention this before, but there are approximately 100 known species of vertebrate from the entire section, right? And that does include reptiles. And they're reptiles and birds, and we also have a lot of very interesting insects. They're not as common as the mammals are, but yeah.
Another question? Just a couple of curiosity because it's not completely.
Of course.
And the first part, I understood that the home classing was a control such as the other one.
Oh, yeah.
No, sorry, you have the control for the classing and non-seven.
Yeah.
That were not directly impacted by the eruption.
Yes.
So just the decrease, as you said, if I correctly understand, is due to the dispersion of species versus, because also in the other habitat, in the control habitats, you saw a reduction of species.
Yes.
And so there is not a direct impact, but it is also only just the dispersion. The hypothesis.
Yes. Yeah.
And I suppose also that the problem with the modern species, not probably to identify them, but to have different scientists making the monitoring.
Yeah.
So I think that both
Something imagine about fossils and the model could have some bias related to
Exactly.
the identification of the species for fossil to go
Right.
on, but
Right.
for the modern, you
Yeah.
cannot have just the squares, they
Yeah.
just monitor or
Yeah.
there's some problem with the sampling and understood, so the
Yes.
fossils for sure I think have in
Yeah.
this, but that's what because also in the second part
Yeah.
the plot after
Yeah.
it seems that before any big eruption there is in some way a decrease of species
Right.
that cannot be related directly to the big eruption. Then there
Right.
is a drastic decrease, but
Right.
I was just wondering if there
Yeah.
could be some answer to this decrease
Yeah.
before the eruption.
Yeah, exactly. No, that's a really good it's a really good point, a really good question. So like it right here, right? We see
Yes,
this.
exactly.
There is another volcanic event between each one of these layers. They're small, so it could be that there is something from that. This one, maybe you see the 1, 2, 3. So it is interesting that you see like, so this one, obviously there's a lot of specimens. This is one of our most diverse layers, but there's not very many species. So it may be that what we're seeing here is that I'm breaking the algorithm for figuring out how many species there are. Because this one has over 500 specimens in it. So it could be that 500 specimens, given the number of species there are, it's actually more accurate than these other ones are. So there could be that. But there are obviously many biases that we have to deal with, which is why I chose this method as opposed to a lot of the other modern ecological methods that we use. And I wanted to use a methodology that would work with the fossil record as well as the modern record. As I was going through and working on this, I noticed the similarities between the way that modern mammalogists collect specimens to how paleontologists collect specimens. And so if I want to use those historic museum collections, I thought this is a perfect way to have something that compares to one another. And of course, that's obviously, standardization in that way is very important. But specifically what's going on in these areas, there could be a lot of explanations. And this is one of those areas where I would need to do a little bit more digging into why exactly things are different in this particular horizon. As far as I know, the geology between these 3 layers here is not any different. There are ashes between them, but they're not very big. So, and maybe there's a larger ecological signal that I'm missing in this particular context. But one of the other problems with these other ones is sample size. And part of that is that these layers up here have a problem of being in very steep areas, which makes them very hard to sample. Or there's only 2 or 3 sites that are accessible. So that does add to some of the difficulty with understanding these layers up here in particular. So that may have been part of the reason why it looks like this. It could be a sampling bias, which I've tried to correct for using the methodology that I used. But we are looking at some of the numbers like here and here specifically of 20 specimens versus this one having 500. Right, so there's these are the problems that we work with the fossil record, but exactly right.
Not the same, but similar.
Yes, one of my favorite. So with the modern ones, one of my favorite ones that I had was a collection of specimens. I believe it was from Washington State in the collections at the Field Museum in Chicago. It was nothing but chipmunks. Right, and it was only the baculum. That was it. Hundreds of little tiny bones, and that was all that I had as a voucher specimen to say that these chipmunks were there. And it was part of a big study that was done to use the baculum as a way to differentiate species in chipmunks. And that's all those data come from, right? So it's like, well, there's bias for you right there in the modern. Like, man, there's a lot of chipmunks. And oh, how big is the fossil? Oh, it's 2 millimeters long, or the specimen. Great, that's all I have. Wonderful.
No, you have done, you have done very good work. It's
Yeah.
really amazing. My
Yeah.
comprehension for the difficulties you are
Yes.
trying to overcome, just in the modern and also in the past.
Yeah.
Dealing just with the one period is difficult, just to compare them with
Yeah. Yeah. I did manage to get the modern one published. So there is an actual paper that goes into more detail on the modern specimens. But I'm still waiting on the fossil ones. But I was going to say, how many hours, Edward, did we spend talking about biases in this stuff? All of the hours. It was a lot. We did a lot. In fact, that was part of the reason why I ended up looking at the old field notes for the modern data to figure out how were they collecting, because we figured out that's probably going to be a big problem with a lot of the modern data. A lot of paleontologists don't do that.
And then you also have the mystery of taphonomy that we explored in class.
Right, exactly.
There are a number of other ways you can measure richness in data like these.
Mhm.
Have you experimented with any of those too, or did you just focus on
I did use a couple of different methods for richness. I don't remember. Oh, I also used evenness metrics too.
Right.
So I had chow richness, raw richness. I also I cannot remember the name of it now. There's another methodology that's in the paper I mentioned that calculates it a little bit differently and uses collection curves to estimate the species or where it should be. So I did look at that one. I got that actually came out in peer review, so I'm really glad someone peer reviewed that and suggested it. But then also the evenness metrics, there were about 4 different ones that I used. And those were the primary the metrics that I used in addition to the NMDS. And
talked a lot about
other distances.
the heliometrics, like with richness metrics and some
Right.
stuff with the two timers.
Yeah.
Did you ever try any of those on modern data, or
I didn't try them on the modern.
metrics are really short? So.
Exactly. So that was part of the reason why I didn't. And even with the fossil ones, those methodologies are designed for much larger data sets. And so I was worried that they would break down under the smaller data sets that the vertebrate record has, because Foote and Alroy were designing these metrics to work with the invertebrate record. And those have, when I have hundreds, they have thousands to tens of thousands of specimens in those data sets. So they weren't as robust in that particular sense. And even the subsampling methods, I wasn't really happy with how the subsampling methods were working in these particular contexts, any of them, whether it be rarefaction or shareholder quorum or any of the other subsampling methods. I wasn't really happy with that, which is why I like these methodologies, because they used all of the data, but then at the same time, they didn't. So I was using all of the data, but they still accounted for everything, and it was one analysis all at one time. That's the statistician in me, right?
Yeah.
One test to answer one question.
Right. Yeah.
Put it all in.
It's huge work and amazing work, and but I think that everybody peer review reading all the work, but you can find, or she can find some problem, but
Of course.
the work is really huge that no one can make criticism
Yeah.
to this, because it's just the effort to try to find out, to
Yeah.
answer question is important.
Yeah, absolutely. Thank you.
Doing, I know you do for 7 weeks or that.
Yeah.
Are you focusing your sampling in one of the layers to improve something?
Well, when I was in graduate school, that's what we were doing. Now we do a much more broader system, and we try to do it more systematically throughout the entire park. Just to try and add, because we have 7 different layers over 40 million years worth of time. So we try to collect everything that we can to answer as many questions as possible. And there are a few sites, and we have done some collections in areas that have bolstered some of those weaker areas. And oftentimes when I've gone out in the field recently, it's been targeting a specific layer because we're looking for additional specimens of an animal. So we actually do have a primate. 1 primate, 3 specimens, that's it. It's not very pretty. We wanted to find more of the primate because we don't know a lot about its ecology. So that was one area where we've gone out and targeted just this summer. And you also never know. So some of these lower layers, like down here, this should actually go up 1 because we just found a tooth to a false saber-toothed cat that we didn't have below that layer before, in that layer. And that was amazing that we found it. It was only one of 3 specimens known of that particular taxon in the whole section. Right. The other thing that does confound this too is that my area is restricted to just that one small area, but there's a large number of specimens that are found in a lot of the other places too. There's the one I'm looking for. So I am up here, and so we're only focusing there in the sheetrock unit, but we also have a site in the southern facies called Logan Butte that has just about as many fossils as the sheetrock unit has. But I had to exclude all of them because the geology isn't quite the same down there as it is up here. So I couldn't. I didn't compare the 2 in this particular situation. Only looking at here, and that's often a question I get too, is like, well, what else could you compare to from the same period of time? It's hard because we don't have the same level of ashes, the same stratigraphy, the same level of correlation between even here and Painted Hills or the southern facies or the western facies. Like, it just doesn't exist in the same degree, though we do have more correlations between the Painted Hills unit and the sheetrock unit than any other place. So we could do a comparison between those 2, but they're in the same species, but also Painted Hills doesn't have the after the ignimbrite collection. It doesn't have that area represented. So it doesn't really work, unfortunately. But I did have a student working on trying to look at heterogeneity across these different sites within these areas. And even within the sheetrock area, which is a very small area, there's very clearly habitat heterogeneity between the different spots. So every spot has a slightly different environment going on, even if it's within like 5 or 6 kilometers of the other sites. So that was interesting to learn about also. And then all of these sites for the plants were doing very weird things. Because that's the other thing we have is a very rich floral record. So we have plants, we have animals, we have fungus, even in one of the sites, we have fossil fungus. So there's a lot there that we can draw from.
Are you planning to check for the plants too? Or are you running mold?
So the hard part is that where there are good vertebrate fossils, there are not so good plant fossils. And a lot of that has to deal with the preservation, the environmental conditions in the rock layers that preserve the fossils themselves. Oftentimes the plants are forming in lake beds, in the lacustrum deposits, but you don't see that happening with the vertebrates so much. So there's a site up here that's a little bit older that has plants, and there's 2 salamanders, a bat, and a frog foot, and that's pretty much all we really know about the vertebrates from that period of time, and maybe a part of a horse, like a shoulder or a hip of a horse, and that's all we have for vertebrates. but then there are 200 plants
Macro.
from macro, yeah. And that's the other problem too, is that those green rocks, the green mineral seladenite forms secondarily, so it forms after the rocks have been kind of put under heat and pressure. And that process of creating that mineral destroys pollen, it destroys the phytoliths. There's basically, we don't know anything about the plants. The only thing that is somewhat useful are the paleosols. To understand the fossil soils are pretty much the only thing that we have that suggests what the plants were doing. Also, obviously, the habitats of the animals. Now, we do get some macrofossils, so at the base of the ignimbrite, we do get trees, but we just have trunks. We don't have anything else to know what exactly it is. We could thin section the wood and figure out if it was metasequoia, which is a Don redwood, or one of the other species, but we just don't have that. We do have a lot of evidence for hackberry trees too, for Celtis or Celtis. They're present throughout the entire, the last 45 million years, we have evidence of them. Even today out there, we have that tree. But that's, we get maybe like 4 or 5 plants in these assemblages, and that's about it, which isn't really that useful.